Animals must defend themselves against many dangerous pathogens they might encounter in the environment. The type of defense is innate immunity and adaptive immunity. Innate immunity for all animals. Barrier defenses is skin, mucous membranes, and secretions. For internal defenses is phagocytic cells, natural killer cells, antimicrobial proteins, and inflammatory responses. For adaptive immunity is vertebrates only. Humoral responses is cytotoxic cells, defense against infection in body cells. Cell-mediated responses is antibodies defend against infection in body fluids. Innate non-specific immunity provides broad defenses present before any exposure to pathogens and effective from time of birth. It provides a defensive capability known as non-specific resistance. For acquired adaptive immunity, it develops only after exposure to inducing agents such as microbes, toxins, or other foreign substances. It's very specific, and recognition is achieved by white blood cells, lymphocytes. Innate immunity in invertebrates. First, immune cells detect harmful pathogens. They extend finger-like projections called pseudopodia to surround the pathogens. Second, the pseudopodia close around the pathogen, trapping it inside the cell through a process called endocytosis. Third, the trapped pathogen is enclosed in a membrane-bound sac called a vacuole, known as a phagosome. Fourth, the vacuole fuses with a lysosome, which contains digestive enzymes. This forms a phagolysosome. Fifth, enzymes inside the phagolysosome break down and kill the pathogens. Sixth, the remaining waste materials are expelled from the cell by exocytosis. Innate immunity, the first line of defenses. First, intact skin, barrier that cannot be penetrated by bacteria or viruses, although even minute abrasions may allow their passage. Mucous membrane, it is an epithelial tissue which secretes mucus. It lines many body cavities and tubular organs, including gut and respiratory passage. Certain cells of the mucous membranes produce mucus. Third is secretion. Body secretions create an environment that is harsh to many pathogens. It contains lysozyme that destroy cell wall susceptible bacteria. It buffs various exposed epithelia by providing washing action that inhibits colonization by fungi and bacteria. In trachea, ciliated epithelial cells sweep mucus and any entrapped microbes upward, preventing the microbes from entering the lungs. There are three types of secretion. First, skin that contains sweat and oils. Give the skin pH between 3 and 5 to prevent any colonization of any microbes. Second, tears of saliva include proteins such as lysozyme that digest cell walls of many bacteria. Third, stomach, acidic environment which kills microbes in ingested food or water before they enter the intestines.
Next, we have innate immunity, second line of defense. In internal defense, we have cellular innate defense, phagocytic cells, natural killer NK cells, antimicrobial proteins, interferons, and inflammatory response. Cellular innate defenses, phagocytic cells. Phagocytic cells engulf many pathogens that defeated barrier defenses of mammals. This is by removing cellular debris and respond to invasion by foreign substances or pathogens. Phagocytes use their surface receptor to attach their prey and engulf them. This forms a vacuole that fuses with lysosome. The two main types of phagocytic cells are neutrophils and macrophages. Phagocytic cells circulate in the blood. They are also attracted by signals from infected tissue, engulfing and destroying infected pathogens, and they tend to self-destruct as they destroy invaders. We have 6 types of phagocytic cells in our body. The first one, we have basophils and mast cells. They release chemicals that mediate inflammation and allergic response. The second one, we have neutrophils. They ingest and destroy invaders. The third one, we have eosinophils. They destroy invaders, particularly antibody coated with parasites. The fourth one, we have monocytes and macrophages. They ingest and destroy invaders' antigen presentation. The fifth one, we have lymphocytes and plasma cells. There are specific responses to invaders, including antibody production. And the last one, we have dendritic cells, recognize pathogens and activate other immune cells by antigen presentation. Next is the process of phagocytosis. First, the phagocytic cell recognizes and attaches to the microbe. The cell then extends finger-like projections called pseudopodia, which surrounds the microbe. The microbe is then engulfed and enclosed inside the cell, forming a membrane-bound structure known as a vacuole. The vacuole fuses with the lysosome, which contains digestive enzymes. This fusion forms a phagolysosome. Inside the phagolysosome, toxic substances and lysosomal enzymes break down and destroy the microbe. Finally, the digested microbial debris is expelled from the cell through exocytosis, completing the process of phagocytosis. Next is the macrophages. It can be found migrating through the body and in various organs of lymphatic systems. It is also well positioned to combat pathogens. The neutrophil has 60 to 70% of WBC. It engulfs and destroys cellular debris or bacteria that invaded the body. It also self-destructs as they destroy foreign invaders. Next, monocytes. They are more effective phagocytic defense of 5% of WBC. It remains in blood for a few hours, migrate into tissues, and develop into macrophages. Other than that, we have eosinophils. It is found beneath mucosal surfaces of 1.5% of WBC. It defends the body against foreign substances or pathogens that are coated with antibodies. Its primary mode of attack is exocytosis of toxic compounds. Lastly, the dendritic cells. It is populate tissues such as skin. The primary role of dendritic cells is to stimulate development of acquired immunity. Next, we're going to be talking about the lymphatic systems. It plays an active role in defending the body from the pathogens. First, the interstitial fluid bathing the tissues, along with the white blood cells in it, continually enters lymphatic capillaries. Next, the fluid inside the lymphatic capillaries called lymph flow through lymphatic vessels throughout the body. Within lymph nodes, microbes and foreign particles present in the circulating lymph encounter macrophages, dendritic cells, and lymphocytes, which carry out various defensive actions. And lastly, lymphatic vessels return lymph into the blood via two large ducts that drain into veins near the shoulder. The next cellular innate defenses are natural killer cells, or also known as NK cells. NK cells patrols the body and attack virus-infected and cancer cells. It also triggers apoptosis in cells that they attack. They do not attack microorganisms directly, but destroy virus-infected body cells. They also release chemicals that lead to cell death and inhibits further spread of the virus or cancer. Next are the antimicrobial proteins, also known as complement system. The plasma contains over 30 complement proteins. Complements are plasma proteins involved in body defense, which circulate in the blood in an inactive state. The complement refers to the fact that this system complements reaction of antibodies. It activated by immune response towards pathogens or their presence. There are three effects of complement activation. The first one is activate phagocytes. Bind to the pathogen so that it can be identified and enhance phagocytosis by macrophages. Next, it triggers inflammation by releasing histamine. And lastly, pore formation causes lysis of invading cells. Next, we have the antimicrobial proteins, interferons. They are small proteins released by activated macrophages and lymphocytes and tissue cells infected with viruses. The actions of interferon, it activates macrophages and NK cells. These activated would these activated would identify and destroy virus-infected body cells. It also would induces nearby uninfected cells to produce substances that inhibit viral replication to limit the cell-to-cell spread of viruses in the body.
Inflammation is localized tissue response that occur when tissue damage or microbes enter the body. Damaged tissue release chemical signals, especially histamine, from mast cell. Histamine causes blood vessels to dilate and become more permeable. This increases blood flows and allow immune cells to reach the injured area. The result such as redness, heat, swelling, and pain occur. Inflammatory response involved in several key events, such as clotting helps begin tissue repairs and prevent microbes from spreading. Second, increased blood flow allows phagocytes to immigrate into the injured area. Third, chemokines attract more immune cells to the site. Finally, pathogens are destroyed by phagocytosis. Innate immunity is a non-specific immunity defense that provides immediate protection. The first line of defense include the skin and mucous membrane, which block pathogens entering. If pathogens enter, the second line of defense becomes active. This includes phagocytes, inflammation, antimicrobial protein, and natural killer cells. Acquired immunity is a specific immune response that develops after exposure to pathogens. It is slower than innate immunity but provides long-lasting protection. There are two types of acquired immunity, that is humoral immunity involving with B cells and antibodies, and another one is cells mediated immunity involved with T cells. Antigens are foreign molecules that trigger a specific immune response. They are usually proteins or polysaccharides found on the surface of foreign cells. Each antigen has a specific regions called epitopes. Epitopes are recognized and bound by antibodies and lymphocytes, initiating the immune response.
Now we look to characteristics of B cells and T cells. A B cell's receptor consists of two identical heavy chains and two identical light chains, and it linked by several disulfide bridges, while T cells receptor consists of one alpha chain and one beta chain linked by a disulfide bridges. How does these two kinds of lymphocytes carry out the immune system response? B cells secrete antibodies that attack antigens, while T cells attack cells infected with pathogens. Now, lymphocyte development. The lymphocyte arise from the stem cells in the bone marrow. The vertebrate body is populated by B cells and T cells lymphocytes which circulate through the blood. B cells and T cell plasma membranes have 100,000 antigen receptors that recognize the same epitope. Here, we're going to talk about how B cell and T cells develop. B cells and pre-T cells arise from the stem cells that originate in red bone marrow. B cells complete their development in red bone marrow, but T cells develop from the pre-T cells that migrate from the red bone marrow into the thymus, where they matured. Before T cell leave the thymus or B cell leave the red bone marrow, they begin to make several distinctive proteins that are inserted into their plasma membranes. Some of these proteins function as antigen receptors. Now is the testing and removal of self-reactive lymphocytes, which is T cells. The antigen of B cells and T cells are tested for possible self-reactivity because lymphocytes with receptors specific for the body own molecules is rendered as non-functional and will be destroyed by apoptosis. T cells were tested with non-self cells and self cells. What is clonal selection? Clonal selection is proliferation of lymphocytes into a clone of cells in response to binding an antigen. Now, this is how clonal selection of B cells happen. Binding of antigen receptor to antigen activates the lymphocytes. And activated B cells amplify, forming two types of clones, which is plasma cells, short-lived activated cells, and memory cells, long-lived cells.
The difference between active immunity and passive immunity. Active immunity occurs when the body produces its own antibodies after exposure to an antigen, either naturally through infection or artificially through vaccination. This process takes time to develop, but provides long-lasting protection because memory cells are formed, allowing the immune system to respond faster during future exposure. For passive immunity is gained when antibodies are received from another source, such as from a mother to her baby through the placenta or breast milk or through antibodies injection. This type of immunity provides immediate protection, but it is short-term because the body does not produce memory cells. What is stress? Stress is the physical or emotional pressure experienced by the body. It affects immunity by causing the release of stress hormone such as cortisol, which suppress immune response by reducing the activity of the white blood cells and slowing down immune reaction. As a result, the body has a higher risk of infection, slower wound healing, and reduced vaccine impact, as seen in the example, such as to the falling asleep during exam and individuals with chronic stress experiencing frequent illness.